For our final talk, I'd like to introduce Dave Gamm. David is a pediatric ophthalmologist who is focusing on the retina in terms of his research. He is a stem cell scientist who is looking at ways that the retina can be reproduced, so to speak, and he will focus his talk today on macular degeneration and stem cells. Dave is part of a very strong group of stem cell scientists here at the University of Wisconsin. Many of you have heard of Jamie Thompson, who's really the father of stem cells and reported on the very first stem cells that were created. That led to some controversy with who should be in charge of creating stem cells and should there be any government regulation. That's still something that you'll hear about in the news. But it is quite a strong team, and Dave has represented ophthalmology in an exemplary way. So thanks, Dave.
Thanks, Barb. That's what we call filler. So I appreciate that. So again, very happy to be here. It's always a pleasure to come and talk to folks. I particularly like to talk about what we're doing at the University of Wisconsin and through our collaborations to groups of patients. I do it quite a bit. The two groups I talk to the most are folks that have inherited retinal degenerative diseases, things called retinitis pigmentosa, which unfortunately cause kids and young adults to go blind over a period of time for which there is really no treatment. And then in the adult population, age-related macular degeneration. So there's a large emphasis on the work that I do towards patients that have AMD. But you know, stem cells, like everything else, have their place. But it's not the answer for AMD, just like no one treatment is going to be a panacea or a silver magic bullet for everyone. It's potentially an answer for some folks with AMD, particularly those who have later stages of AMD, but it's not the answer. And I'll also mention right off the bat that you're better off if you never need a stem cell, right? I mean, so you're always better keeping what you've got than replacing what you've already lost. So the efforts that you heard Dr. Maris mention and other people in the realm of prevention of AMD is the way to go if at all possible. If not, if you fall through the cracks and you do end up having an untoward result with AMD, then stem cells may very well bring a form of therapy to you in the not too distant future. So that's what I'm going to talk about a little bit today. So as Dr. Bodie mentioned, I'm a pediatric ophthalmologist, and all ophthalmologists, and frankly all doctors, for the most part, only like to deal with things that they can help. It's not easy for folks to just diagnose and say, well, there's not a whole lot we can do for you. So what I'm showing you here are just some various cases where you're talking about, in my field, various growths and malformations that can be removed. If the eyes aren't properly aligned, we can align them. I was doing that this morning. Cataracts can be taken out, and foreign bodies and injuries can be oftentimes fixed. But when you talk about AMD, there's a lot of times that there's not a whole lot that we can do about it. And the mission of my laboratory is really to use stem cell technology, which was born here at the University of Wisconsin and has subsequently developed, and we're really one of the major centers in the world in this field, to use this to address folks that have blinding disorders. And one of those blinding disorders, as I mentioned before, is macular degeneration. And so on the top of the slides here, what I'm showing is on the left is a normal retina with a normal, kind of in the center, you see that more yellowish area, and that's yellow, and you know why it's yellow now from Dr. Maris's talk. And on the left, you see that it doesn't look quite the same. In the center, there's this kind of amorphous, amoeba-like thing that looks more yellow than it should be. And that's actually an area of macular degeneration that's causing death of critical cells in the retina that you need to detect light and transmit an image to your brain. So ultimately what you see in a normal person, if you're looking at these two boys with balls, they say, okay, looks nice, it's pretty pleasant. But in the patient that has that central area of atrophy or degeneration, that center portion is gone. And of course, that's what we use them to read books, that's what we use to recognize faces, and so it's really critical for us. And we lose a lot of our mobility and our independence when that central vision goes away. It's important, and somebody's probably already mentioned this to you, that you don't go blind from AMD. It robs you of your central vision, but not the peripheral vision. So if that's all you have, you're not going to go completely blind from AMD. And I think a lot of folks do have that worry and that concern. So to discuss a little bit about where stem cells lie in the continuum of possible therapies for AMD, I want you to to give you a little bit of the basic biology of exactly what goes wrong in the back of your eye in AMD. So the retina is, and if we take a look here, kind of slicing the eye in half in a non-gory way, and then looking at it from the side. So we're going to look at a cross-section of the eye. And to the left here, you see where the light comes into the eye, the cornea, the lens. It passes through the jelly that fills most of the eye, called the vitreous, and then strikes the back of the eye, where there's a film, a biofilm, so to speak, that's called the retina. And it's very thin. I mean, it's transparent to look at, but actually it's a very elegant layer cake of cells. And each layer has a different type of cell, and they all do different jobs. And they connect in a very certain way that allows light to be transmitted, light information to be transmitted from the deepest cells within that tissue, called photoreceptors, and then eventually go through this kind of arc back to the brain. So the two cells that I'm most interested in the retina are the two deepest layers. The photoreceptors, which are shown here in red, which are the rods or cones, these are the light sensors of your eye. And then directly beneath them, or next to them, is a layer called the retinal pigment epithelium, and or RPE, which is what I'll refer to them as. And the RPE are the handmaidens of photoreceptors. Photoreceptors get all the credit. They're the glamour cells of the retina, but they can do very little on their own. They're just like all divas. They can't dress themselves. They can't do anything. So the RPE are the cells that actually do all the work, make sure they get fed, make sure that if they throw their wrappers away, they get picked up and moved away so garbage doesn't collect. So that kind of Batman and Robin sort of arrangement with the photoreceptors and the RPE is critical for the initiation of vision in the retina. And they get connected. They don't sit there by themselves. They get connected, and they're really neurons, just like in your brain. So the retina is actually just a portion of your brain that's more external and is specifically designed to detect light. So those photoreceptors connect to bipolar cells, which connect to another cell called a ganglion cell, which are involved in glaucoma when they die, and then they connect back to the brain. So it's a relatively simple kind of mini brain that's specifically designed to detect light that lines the back of your eye. Light goes all the way through that transparent layer cake, strikes those photoreceptors, and it relays a signal back to the brain. And then the brain is what actually does the seeing. It puts those signals back together. So in macular degeneration, the focus of the disease is really on the helper cells. Okay, it's more complex than that, but for the most part, you got to think about those RPE cells, those handmaiden cells, are the ones that take at least the first punch. And so what happens is they will become dysfunctional and eventually die. And when that happens, the divas aren't long for the world because they can't feed themselves and they can't clean up after themselves. And once the photoreceptors go, you lose the ability to initiate that sense of, I see something, okay? And then you get the blindness. Another thing that can happen, and if that's what happens and that's all that happens, it's called dry AMD. Sometimes, though, when that RPE goes away or degenerates, the blood vessels that are coursing behind it can grow up into the space where the RPE used to be next to the photoreceptors, and they can break and bleed, and that causes the wet type, and it will cause a more kind of rapid acceleration of photoreceptor death. So the main thing to take away from all that is that we're talking about loss of cells. Your retina, just like your brain, has no capacity to replace its neurons, its critical cells, on its own. So some things in your body can do that, right? Your liver has a low ability to regenerate itself. Your blood does continuously through your life. Your skin does. If you have a scratch or whatever, it'll grow back in. But your brain and your retina cannot. So there's no intrinsic mechanism to repair damage to those cells once they're gone. So if they're dead, they're gone. You can't make more of them on your own. So what we're doing is we're missing parts. And so what stem cells then provide is, at least theoretically, a source of spare parts for human cells that are lost during degenerative processes that don't regenerate on their own. One of those being the photoreceptors. So stem cells also have a couple other unique properties. One is that they're able to divide indefinitely. So they're an infinite supply. You don't run out of them in a dish. So once they're made, they can be made and propagated theoretically forever. And then they can go on to make all the different cell types in the body. That's a good thing and a bad thing. It's a good thing because, again, theoretically, you can make any cell type in the body. And so for those cells that don't regenerate themselves, you then have perhaps a source, an unlimited supply of those spare parts. The downside is that they will make every single cell in your body nearly. And if you don't control that, you'll have perhaps a photoreceptor around a bunch of bone, around a bunch of beating heart cells, and so on and so forth. And you can't stick that whole commission to somebody's eye. So you have to understand the process and control it. And that, like a lot of things in science, is the hardest thing to do. So when I talk about stem cells, what are the sources that I'm talking about? So you hear a lot about, oh, stem cell this and stem cell that. There are stem cells in your bone marrow, and they've been used in bone marrow transplants for decades and decades. But they only make blood, so they're good for patients who need a replenishment of their blood cells after, say, treatment for leukemia or something along those lines. But those blood stem cells can't make eye, they can't make brain. They're specifically designed to make blood, and that's what they'll do. So sometimes people will say, well, what about those stem cells that you give to leukemia patients? Well, they're not going to help a person who's lost retinal cells. So people use stem cells very, very loosely, but there are dozens and dozens of different types, and they're not all created equal. But especially some of the shyster companies and organizations that you'll find on the internet, they'll just try and confuse you by saying, well, we'll give you a stem cell. And they won't tell you what kind, because everyone thinks stem cells are magical, which they're not, and that you'll break out your retirement fund and pay them to do it, right? And this happens, and it happens in the United States, and it's not just something you have to go overseas for. But the only two types of stem cells that can make eye cells are what are called pluripotent stem cells. The original type were embryonic stem cells. These are the Bush lines that were established, but now going on 2 decades ago. And those are the initial ones that were made. And another type are called induced pluripotent stem cells. Now, or iPS cells. Now, iPS cells are different. They're actually adult cells taken usually from blood or a blood sample like you might give for a cholesterol check, or a small skin biopsy. And then those are reverse engineered genetically back into an embryonic stem cell state. And then from there, we've developed techniques to forward engineer those to people's actual retinas. And so my laboratory uses almost exclusively iPS cells because we can make it from any living human being on the face of the earth, and they're unlimited supply. So here's an example here of a dish. So these are my hands here, and you see 6 little wells here. And it looks like they're kind of dusty and black, right? So what these were initially stem cells. This was initially a blood sample from a patient, which we then engineered into iPS cells, and then adopted one of our protocols to forward engineer into RPE, those retinal pigment epithelial cells that are supporting photoreceptors in a person's eye. So these are billions of cells from one particular patient's. And so if you look more closely, this is what they look like. And they look like these little tiny, each one of these little circles, these little octagons that you see here is an individual RPE cell. And we can make billions of them from any one particular patient. And these do all the things RPE should do. They can do all the handmaiden stuff. They can pull garbage away, they can provide nutrients, and so on and so forth. The thing is, they're in a dish, they're not in somebody's eye. And so this is actually a family of actually the woman on the left in green, the gentleman on the right in blue, are the 2 people that we made the stem cells from and ultimately made the RPE from. The woman on the left doesn't have any diseases. The gentleman on the right has a particular disease that causes a type of macular degeneration. And so I'm going over one of our studies that grew their cells, and we used them to learn a lot more about his disease and to screen for drugs that might slow it down. So that's using them to perhaps screen for drugs and things like that to slow down disease. But actually, these same cells are now being put into patients. So it was one of the first clinical trials that was initiated was for replacement of RPE in AMD. And there are now 2, going on 3, and soon 4 clinical trials to do that. And so right here, this is a friend of mine, Dennis Clegg at UC Santa Barbara, who's involved in a large California project that I collaborate with. And they've seeded those RPE cells on a special little, looks like almost like a lollipop. And it can be folded up and placed under the retina in a patient with AMD. And it's been safe thus far. And it showed, some modest effect. But the issue with it is that, as I mentioned before, in AMD, you lose the RPE cells first, right? But then you lose the photoreceptors. So it's like losing pistons and a carburetor. You got a carburetor on. And in this case, we're only replacing one of those spare parts. So it's limited what you can do with RPE by itself because you haven't produced the photoreceptors and included those in the avenue in which you're trying to treat the patients. So it's one of the parts, but not both of the parts. And here's an example in a retina of one of these little lollipops that have been inserted under somebody's retina. It's black and white. You're used to seeing the color ones, but it's a black and white version. And the darker gray lines are blood vessels. All right, so what about photoreceptors then? So RPE cells are one thing, but it's only half the battle. The other half are photoreceptors, which are much more complex and difficult to make. And they have to do a lot more things, and they're fragile, just like divas are fragile. So here you see a bunch of different photoreceptors, particular ones called cones, which are the most important type of photoreceptors in the macula that are lost in AMD. And so what we developed is we developed a technique to actually grow whole retinas, little mini retinas in a dish. So we can take those same blood samples, those same skin samples, reverse engineer them to stem cells, and then forward engineer them into these little mini retinas. And we can make, again, an unlimited supply. So each one of these, we call them these golden Cheerios, is a developing retina from an individual. And if we cut it in half and look at all the cells that are in them, if we look really early on, this big donut, blue donut looking thing, every little blue dot in here is a cell, and every little red smudge that you see is a photoreceptor cell. And it grows and expands and develops just like they would in a developing eye in utero. And over time, eventually, we see even these little tiny little hairs that are on the outside of these structures, and they grow to be about, maybe 1/2 millimeter or so. So by no means are they the size of our eye. They're all on a mini scale. But we can then dissociate and put them together and kind of make them larger if we need to. When we do that, we find that as they grow, they actually grow and laminate, form those layers just like a regular eye would. In the outside we see, and those red dots are cones. The purple dots that are beneath that are all rod cells, so they actually know what layer to be in. The cones know to be on the outside, the rods, know, which detect black and white, know to be in the middle, and the other layers of the retina are still there too, although we have ways to get rid of them because, again, we only want the outer portion. So by itself, we can grow those like individual little balls of retina, which gives us the spare parts, the photoreceptor spare parts, which we're now trying to meld with the RPE to get a more complete therapeutic product for folks with AMD. But that's not easy to do. We've got RPE growing over here, we've got photoreceptors growing over here. How do we kind of get them to mesh together for one thing that could be easily inserted into somebody's retina with the hope that it would then connect and restore vision? And to do that, we then have to look elsewhere on our very busy and very expert-laden campus at the University of Wisconsin-Madison. And for that, I look to our bioengineers, because a lot of what we do is not just a biology problem, it's an engineering problem. And so we've been able to develop these little scaffolds, and each one of these little holes in these scaffolds can fit one photoreceptor cell. And we can make these little molds where we can have large sheets of these, and they can be cut into a very specified shape for an individual patient. Oops, let's go back. And like I said before, each one of these little wells can hold one photoreceptor. Sometimes they hold more than one. And the example I'm showing you here is each one of the little red lines is an individual photoreceptor that's sitting down in one of these wells, and they're oriented in a proper way so that they can interact both with the RPE cells that need to help them, as well as the other cells they need to connect with to send signals that they've detected light and that the brain needs to perceive an image. So we're at that point, now we're at a point where we're working with industrial partners to do this in a fashion that's safe for human use, and that's a difficult thing to do because we have to take everything that we do in the lab, and every little part of that has to be made very efficient, safe, reproducible across multiple different stem cell types in order for us to be able to convince the FDA that this is something that we're first going to do no harm with and has some capacity to help a patient. But it's important to remember, too, that we didn't get, the Wright brothers didn't stand around and say, boy, I'd like to be able to fly, and then the next day had a jumbo jet. Folks, they had to have some things that were pretty rudimentary first, and then they had to figure out what worked, what didn't, and then over the course of years, the technology developed, and we had something that really was amazing for humankind. Stem cells, I think, are going to be the same way. These are the initial steps that we have. We have to be able to make the spare parts. We have to be able to do safe studies, hopefully get at least modest results, find out what works, what doesn't, change things appropriately, and then ultimately get there with version 2.0, 3.0, maybe 20.0, where we're really helping patients to see better and improve their standard of life. And so with that, I'd like to thank you very much for your attention, and I will step aside.